everyone. I'm going to be doing my presentation on the isolation streak plate technique lab that we did. My name is Shannon Sipes. This is for Life 1412 Microbiology, and it is the fall 2024 semester. All right, so jumping right into it. So when discussing microbes, it is important to understand the difference between pure and mixed cultures. So a pure culture contains a single species of microorganism that is genetically identical. This means it's free from contamination from other microorganisms and can thus be studied for specific properties or be identified in a laboratory setting. In contrast, when we're talking about a mixed culture, these contain two or more different species of microorganisms in the same sample. So when it comes to real world scenarios, we're more likely to encounter mixed cultures from natural environments, and these can contain a variety of bacteria, fungi, and other organisms. To be able to separate out the organisms present in a mixed culture, you must be able to utilize something like an isolation streak plate method, which is what we did in this lab. So an isolate streak plate technique involves methodically spreading the mixed culture over the surface of an agar medium. This allows for separation of colonies, which are distinct clusters of cells, that's what colonies are, and are thus then isolated and are now pure microorganisms. So essentially what you're doing is you're grabbing a loop of inoculum and you're dotting it onto the agar surface aseptically. And this is then dragged in a zigzag pattern in one of the quadrants on the agar. So if you're taking a look at this, this is kind of showing what I'm going over. So then what you're going to do is you're going to grab a new sterile loop, and then you're going to use that to drag some of the inoculum from the original section to the quadrant right beside it. This process is then repeated until there are 4 quadrants of streaking completed, and then you're going to incubate this. So if you've done this correctly, then you're going to be able to see that growth will occur and the colonies will be isolated as the inoculum streak becomes more diluted across the plate. So when you're looking at these, I wanted to show you kind of a representation of what a pure colony might look like versus a mixed culture. So for pure cultures, as you can see here, even though it's been streaked out for isolation, there is only one red colony, so it's not mixed. So this is what a pure culture would look like. As far as a mixed culture goes, this is kind of more of that representation. You can see distinctively there are 2 different species here. So we've got one that's more yellow and then one that's a little bit more opaque. All right, so for the objectives here, the first one is you're going to want to be able to utilize the isolation streak plate technique to derive pure cultures from a mixed sample. You're also going to be able to identify and define the concept of a microbial colony and what it might look like on an agar medium. So as I was saying earlier, colony is basically a cluster of cells. And colonies can have a lot of different characteristics. So this might include color, this might include whether or not it's convex or concave. It can also be matte or glossy. So there's a lot of different ways that a colony can present itself, and this might help you be able to differentiate a pure versus a mixed culture. So that then kind of leads us into this third point, which is to be able to explain the distinction between a mixed culture and a pure culture. So moving on. All right, so the things that you will need for the lab is you're going to need proper PPE, so this includes your gloves, your lab coat, your safety goggles. You're going to need inoculation loops, sterile plastic pipettes, an empty sterile tube. You're going to need a marker, so this is what we're going to be marking our plate with, just so we know what's in it. Flame source, this can be your candle and a lighter. You'll also need a culture rack, 2 tryptic soy broth tubes, your micrococcus and your serratia. You'll end up needing to reactivate those, but you'll need those. You'll also need your tryptic soy agar plate, a surface disinfectant, disposal jars filled with 70% IPA, and disinfection container. All right, so as far as the steps go, the very first one, we're going to gather all of our materials, wash our hands, and dress in our proper PPE. So this is pretty much how we start any of our microbial lab experiments. You're also going to be disinfecting your workspace and then igniting your flame source. So for me, I had used the candle, and I used the lighter to go ahead and ignite that. Third step, we're going to be reactivating the cultures. So this is going to essentially mean we're going to be aseptically transferring 1 milliliter of tryptic soy broth from a tube into the vial that has the culture pellet in it. So for example, we're obviously going to be doing this one species at a time. So say we start with the Micrococcus, we'll be doing transferring 1 milliliter of the tryptic soy broth into the Micrococcus vial. We're going to wait 30 seconds, and then we're going to aseptically transfer the contents that were put into that vial back to the tryptic soy broth tube that the broth came from. We're going to now allow this to incubate for 2 to 4 days until growth is present. So this you'll then, once you do it for Micrococcus, you'll go ahead and repeat that exact process for Serratia. Once there has been growth, and you'll know this by basically seeing some turbidity or, sort of some disturbance in that broth tube, then you're going to go ahead and proceed to step 4. This is going to mean you're going to roll or tap the culture tubes to resuspend that bacteria, and then you're going to aseptically transfer 10 drops of your Micrococcus and your Serratia to a sterile culture tube. This is essentially mixing them up so that we can get a mixed culture or a representation of a mixed culture. So then you're going to tap that culture mixture just to make sure that it's all mixed up properly. And then the next step, step 6, you're going to be utilizing a sterile inoculation loop to obtain the mixed culture broth. You're going to inoculate 1 quadrant of the tryptic soy agar plate using a zigzag pattern. And then you will take a new sterile loop, streak from the first quadrant, so you'll overlap it a little bit. You'll streak from that first quadrant that we added bacteria to, and then you're going to be streaking it into a new quadrant right beside the first sector, also using a zigzag pattern. Step 8, you're going to be repeating this process, which are step 6 and step 7, until all 4 quadrants are streaked. And then at that point, once everything's streaked, you will incubate the plate lid side down. Again, we're going to mark on here, if you haven't already, what the culture actually is. So we can just say our mixed culture of Micrococcus and Serratia. You're going to be incubating this for approximately 96 hours total, and you'll be checking it at hour 48, 72, and 96. And then our last step, as always, we're going to be properly cleaning up our workspace. So that might look like we're going to be taking all of our inoculation tools, like the loops and the pipettes, and we're going to soak those in IPA for 30 minutes, then pour the IPA down the drain and put the tools in a plastic bag in the trash. As far as the cultures and the vial containers go, we're going to be disposing of those in 10% bleach bath for 24 hours, and then once the 24-hour mark is done, we're going to pour the bleach down the drain, and then the items are going to go back into a plastic bag and then into the trash. At the very end, you'll disinfect your work surface and wash your hands. Alrighty, so just kind of talking about the results. One of the biggest ways that you can kind of see if you were successful, like I had mentioned earlier, is kind of taking a look at the colony morphology and being able to see if you were successfully able to separate them. So for this experiment, the color difference between the 2 bacterial species is incredibly helpful because it allowed for me to easily see if I was able to separate them correctly, aka isolate the colonies from one another. So looking at the beginning of the streak, you can see that it's difficult to see what colony types were present as they were more like one on top of another. When looking at the end of the streak, you could see that 2 different colonies are, separated distinctly because of their color differences. I could accurately point out which colonies were Serratia, because those were more red or pink in color, versus the ones that were Micrococcus, which had a little bit more of a yellow color. Because of this, it signified that the experiment was performed successfully. If, however, say the bacterial species had the same color colonies, you would then have to use other methods. So I had mentioned earlier talking about colonies. and a little bit about morphology type. So examples of this would be scientists being able to differentiate bacterial colonies by colony size. So maybe one's bigger or smaller than the other, the texture, elevation, form, and the margin of them. And I, of course, mentioned color as well. So that's going to be kind of the results from this lab, but going forward, I'd like to talk about sort of factors that influence these results. So when looking at my results from the lab, I believe there should have been a better distribution of growth among the 4 quadrants than I ended up with during my experiment. And there are a few factors that could have impacted this and in general can impact isolation techniques. So this would basically be your technique, your inoculum density, agar temperature, and lighting. So with isolation techniques, this could mean the streaking technique might not have been consistent in each quadrant, or that there was an insufficient amount of bacteria spread into each quadrant. As far as inoculum density goes, that could mean that when you're placing this inoculum on the agar, you could have put too much or too little. So if you put too much, it might result in too much growth and no isolation. But if you put too little, there might not be very many colonies for you to work up with the isolated colonies. So another factor that could have affected the results would have been proper lighting. So if you're unable to see your previous streak lines, it makes it very difficult for you to drag inoculum from the previous quadrants. And then additionally, the agar temperature could have also affected the outcome of isolation techniques, because if the agar is too cold, it can alter the consistency of the agar and make it more difficult for colonies to develop properly. So for my own results, I believe I could have improved in a couple ways. I could have monitored the inoculum density a little bit more closely and made sure that the agar was also brought to room temperature, because when I was plating it, I had noticed that it was still a little bit cold to the touch, even though I did let it sit out for 15 minutes from the fridge before I started. But it still was cold. And I also think I could have improved on the lighting conditions, because obviously, if you're not really able to see your streaking, it makes it a little bit harder to do this properly. So I think by addressing these factors, I'd be able to achieve a more effective isolation of the bacterial colonies in future experiments. All right, so the broader importance of this. My lab results contributed to a broader understanding of microbiology in a couple of different ways. So firstly, they demonstrated that you can isolate individual bacteria species from a mixed sample. And this is very relevant in a lot of different microbiology fields because, as I mentioned, pure samples are rare in nature, and more than likely you're going to get, most organisms coexisting with various microbes, including bacteria. So isolating these species enables scientists to distinguish between, say, normal flora and potentially pathogenic bacteria. And this can be crucial in a medical setting because it can allow for accurate antibiotic prescription. So essentially, my results highlighted the unique colony morphology of different bacterial species. And as I said, this is very essential for identifying them in a laboratory setting. And can allow for technicians to analyze diverse patient samples to diagnose infections. So I kind of wanted to mention like a real world example with this. So for example, if you have a patient who comes in with a symptom of a urinary tract infection, they might require culturing to identify what species is involved, what bacterial species is involved. And it's possible that they might have 2 different strains of bacteria present as well. So when you get this, if you're able to isolate those colonies by using this technique, you can then separate and examine each type of bacteria and this would allow for susceptibility testing on individual strains to be performed. And that might mean that each species requires different antibiotic treatment. So this is very valuable information, and it enables a more tailored treatment option based on specific bacteria present. So I actually work in a microbiology patient testing facility, and this technique is very applicable, and we use it every single day. So it's very important. Lastly, I just wanted to mention my references that I used that helped me put together this presentation. and say thank you very much for watching. I hope you have a great rest of your day.